Assalamu alaikum everyone and welcome to Ordinary Days. In this video we will discuss the chapter number 8 from your lippincott, that is the introduction to metabolism and glycolysis. Now, what is metabolism? It is sum of all the chemical reactions that are occurring inside the cell at any time. And these metabolic pathways, they can be divided into catabolic pathways and anabolic pathways. The catabolic pathways are degradative pathways that are going to break down the complex molecule, whereas anabolism, as you know, when you were kids, you used to study this catabolic and anabolic thing. So anabolic pathways, they are synthetic ones that make the complex molecules. Like the breakdown is breakdown of like proteins, etc., whereas the synthesis pathway is making of proteins from the amino acids. So now catabolic pathways, they produce ATP by breakdown of larger molecules. What are the larger molecules? Like proteins, like carbohydrates, like fats, etc. And they are generally oxidative reactions or exergonic reactions, and they have three stages. First stage is hydrolysis, second stage is conversion of the products to acetyl coenzyme A, and then we have oxidation of acetyl coenzyme A. If we look at the book, you can see literally the stage 1, which is hydrolysis of complex molecules. Let me zoom in. So this is hydrolysis of complex molecules like proteins, carbohydrates, fats. They are hydrolyzed into the amino acids, monosaccharide, and the glycerol and fatty acids. So first is hydrolysis. Hydrolysis produces these byproducts. And second step is the conversion of these smaller products into acetyl coenzyme A. And third is the oxidation of acetyl coenzyme A in the tricarboxylic acid cycle to produce ATP and carbon dioxide. So this reaction or these catabolic steps, they produce ATP and they are exergonic or energy releasing processes. Whereas the anabolic pathways, they use ATP to make the complex molecules. That is why they are endergonic reactions. They require ATP and they require heat to make them happen. And catabolic pathways, they were oxidative ones. So anabolic will be reductive pathways, and electron donation or reduction, this hydrogen will be given to them to reduce these steps by NADH majorly. Now we will discuss the regulation of metabolism. It is a very important topic, and there are four mechanisms by which we can regulate metabolism. It can be regulated by intracellular communication, by intercellular communication, by G protein linked receptors, the second messenger pathway, or by adenylyl cyclase. So first is intracellular communication, or you can call it the communication that is occurring within the cell. It is for moment to moment regulation. It is like it depends on availability of substrates, the product inhibition, allostatic activators or allostatic inhibitors. Now intercellular communication, that is a communication between the cells, and it is a long range integration or long range interaction. And there are three things that majorly regulate this pathway: the intercellular communication, that is via synaptic signaling, via endocrine signaling, and via direct contact. So what is synaptic signaling? Let me go to the book. So as you can see, this is a synaptic signal. So it is a neuron or nerve cell is connecting to another target cell by means of neurotransmitters. So this is synaptic signaling. The other one is the endocrine signaling, which is by the means of these hormones to the target cell. And third one is cell to cell direct contact by the means of these gap junctions between them. So it can either be from a nerve cell to a target cell by means of neurotransmitter, from a blood cell to another target cell by means of hormone, and from one cell to another, generally closely placed cells, by means of this gap junction. Now the third thing that we will discuss, the first two things were the intracellular communication, that was inside the cell, inter between the cell. Now G protein link receptors. Now we have receptors on our cell, whenever your substances like these neurotransmitters, these hormones, they are coming, they are going to attach to a cell on its receptor, and that receptor is further attached to a GTP binding protein inside, and that GTP binding protein then activates a second messenger system. Now we have various type of second messenger systems. We have phospholipase C system, which depends on calcium. We have the adenylyl cyclase system, as you can see over here, and we have the phosphatidyl inositol system. Now phospholipase C, phosphatidyl inositol, and adenylyl cyclase system. Now, in all the three systems, what is major thing? What first thing is reception, binding of the ligand to the receptor is reception. Then transduction. Transduction is actually the mechanism through which the intracellular domain connects to the transmembrane domain connects to the extracellular to the intracellular one. That is transduction, and then there will be a production of response. So, what will happen? Firstly, there is ligand binding, then there is transmembrane protein activation, and then intracellular alpha, beta, or gamma subunits are activated. What do I mean by this? Let me show you. So, if you look at your book, you can come up to this. This is the extracellular portion, this is the transmembrane portion, and this comes out to be the intracellular portion. Now, this extracellular portion is ligand binding domain. This transmembrane portion is the reception or not reception, most probably it is called transduction domain. So this attachment, first one was reception, second one is transduction via the transmembrane thing, and third one is production of the response by means of this intracellular domain. Now this intracellular domain is attached to certain types of things. They are alpha, beta, and gamma subunits. So in case of this adenylyl cyclase, we can see these receptor intracellular domain bound to this alpha, beta, and gamma subunits. Now, in the next thing, we will discuss the adenylyl cyclase. Now, your adenylyl cyclase, it is a very important type of thing, and it is generally activated by alpha 2 and beta receptors, which are adrenergic receptors. Now, first thing is the G protein activation. Now, let's have a look from the book. So if we go to this first diagram, let me zoom in a little bit if we can. So first diagram, as I told you, that this was the receptor extracellular domain. It is having a hormone which is going to attach to this receptor. Whenever this hormone will attach to the receptor, it will activate this alpha, beta, gamma subunit thing, and then it will act on this adenylyl cyclase. So what do I mean by this? First of all, this alpha, beta, and gamma unit, it is attached to GDP and it keeps the cell inactivated. But as soon as the hormone it binds to this receptor, what will happen? Now GTP will come and it will attach to this alpha subunit. And whenever GTP will attach to alpha subunit, it will separate from this beta and gamma subunits. Now what will happen with this? This alpha subunit is now going to go to this adenylyl cyclase, activate this adenylyl cyclase, and this adenylyl cyclase will convert the ATP into cyclic AMP. ATP, as you know, it contains 3 phosphate groups. So when the ATP only has one phosphate group, it is known as AMP, and because it is a cyclic form, so it will be cyclic AMP, and the 2 remaining phosphates, they are connected together, so they are inorganic phosphates. So what was the thing? Attachment of the hormone, then GTP binding in place of GDP, and then that GTP causes alpha units to separate. This alpha unit now binds to this adenylyl cyclase, converts ATP into cyclic AMP and inorganic phosphate, and that after that, what will happen? As soon as this whole pathway is done, what will happen? The GTP that was attached, now it is going to convert into GDP. And whenever this GDP is attached to that alpha subunit, it will now again attach to beta and gamma subunit. So they will again become brothers and they will exist as alpha, beta, gamma combined subunit with the presence of GDP. But whenever there is GTP, what will happen? The alpha subunit will disintegrate. Now this was the first step, that was the G protein pathway. After that we have the protein kinases. So now what does this cyclic AMP do? This cyclic AMP is going to activate protein kinase A. So if we look at this diagram of the book again, so this is our cyclic AMP dependent protein kinase A. which is having regulatory subunits and catalytic subunits. As soon as the cyclic AMP that was produced from adenylyl cyclase, it is coming to this much protein, it will separate its regulatory and catalytic subunits by binding to its regulatory subunits. When this catalytic and regulatory subunits are separated from each other, what will happen? That this catalytic subunit will now convert the protein substrate, or it will catalyze it, or it will phosphorylate it. So what was the thing? Cyclic AMP coming, separating the regulatory and catalytic subunits by binding to the regulatory subunits. Now catalytic is free. What will it catalyze? It will catalyze the phosphorylation, convert the protein that was non-phosphorylated to attachment of the protein, that is phosphorylation. But after phosphorylation, after the thing that has been done, we have protein phosphatase. The protein phosphatase has the activity that it can remove this phosphate group. And how it does the removal? By adding water. It will be again dephosphorylated and protein substrate will go back to that. Now this whole thing that was why was this phosphorylated protein required in the first thing? It was required to produce intracellular effects, but as soon as the effects are not needed, or after a particular time, the phosphatase is going to act, it will remove this phosphate group and we will get our original dephosphorylated protein. Now what will happen to that cyclic AMP which was produced over here? The cyclic AMP, it will be hydrolyzed. It will be hydrolyzed in such a way so it becomes ineffective. So cyclic AMP will be converted into 5' AMP, which is like it has no important function. So it cannot activate the cell in the way cyclic AMP did. So this is done by cyclic AMP phosphodiesterase, which is going to do the hydrolysis. Now this cyclic AMP phosphodiesterase is actually inhibited by caffeine or methylxanthine derivative. Why is caffeine so important in this reaction? Because caffeine, when we take caffeine like coffee, like tea, it is going to inactivate, or it is going to stop the conversion or stop the breakdown of cyclic AMP. And whenever the cyclic AMP is produced in more amounts, what will it do? It will always support the catalysis, and whenever catalysis is supported, what will happen? Intracellular effect will be produced in large amount. That is why we are activated whenever we take coffee and tea, that are methylxanthine derivatives that will inhibit the cyclic AMP hydrolysis or its deactivation. Understood? Now we will talk about certain diseases related to adenylyl cyclase. Inappropriate activation of adenylyl cyclase leads to two types of diseases. One is Vibrio cholera and other is Bordetella pertussis. So Vibrio cholera, the normal cholera, what will happen in this situation? GTPase activity of the alpha stimulatory unit is inhibited. And what will happen in such a situation? Adenylyl cyclase is, they will be inactivated. It will be inappropriately activated. How? By the inhibition of activity of G alpha stimulatory system. And in Bordetella pertussis, whooping cough, the inhibitory unit, it is inactivated. When inhibitory unit is inactivated, what does it mean? It means the stimulatory unit is doing its all function. And in both the situation, adenylyl cyclase will be super activated, and in Bordetella pertussis or whooping cough, you have a large amount of cough, you have respiratory tract cells, they are filled with mucosa. Why? Because adenylyl cyclase is produced in a large amount. In Vibrio cholera, you have a large amount but of feces. So with that, we are done with introduction to metabolism and glycolysis first part. In the next video, we will discuss, or we will start with the glucose transport into the cells. If you like my videos, if you like my notes, please like, share and subscribe, and you can take a screenshot if you want to. The whole chapter is, the whole 3 to 4 pages, they are summarized in this one page. Allah Hafiz. Let's meet in the next video.